Okay, so happy Tuesday, everyone, and thank you for joining us for another installment of the Paleo Perks seminar. And today we are joined by Savannah Olroyd from Utah Valley University in USA. And today she will be talking to us about exploring the mechanisms underlying the convergent evolution of heterodonty in vertebrates. So before we go any further, we'll quickly go over today's format of today's seminar. For those of you who are new or joining us after a long time, we are having welcome announcements right now, which will be followed by the talk, which will be followed by the moderated question and answer session. So don't forget to send your questions via chat to the questions at PaleoPerks host, who today is Nina. And then our speaker has kindly agreed to be with us for tea time. So anyone is encouraged to join during that session to have an informal chat with our speaker. A bit of housekeeping. Paleo Perks values the participation of all folks interested in the paleo sciences. Please remember to abide by our code of conduct during today's seminar. You shouldn't be able to unmute yourself, but in case you do, please mute yourself for the duration of the talk out of respect to our speaker. You can ask questions by chatting to the questions at PaleoPerks host or by using the raise hand function. If you face any technical issues during the Zoom, please do not hesitate to reach out to the questions at PaleoPerks host. We also have closed captions built into Zoom. Use the CC button to show or hide them. Radio Perks works on the nomination of outstanding early research careers, um, like our speaker today and most of our participants. So please, we encourage you to nominate an outstanding paleo researcher that you know of. We also collect anonymous weekly feedback to uh, be informed about the demographic of our audience. And this form is optional, but you're encouraged to also fill this form. Both the links to the forms have been dropped into the chat. Um, and now to introduce our today's speakers, Savannah Olroyd. They did their bachelor's in science from the University of Oregon. Then they did their PhD in the University of Washington, USA, and now currently they are assistant professor at University of Utah Valley, USA. Savannah, please share your screen and begin your talk.
All right, great. Thank you. Uh, Let me just share my screen here. All right, cool. Thank you all for joining me on what for me is a bright and sunny morning. Definitely one of the benefits of moving to the desert. I'm going to be talking about convergent evolution and some of the things I find interesting about it and some of the things that I am trying to research in my lab here at UVU. Let me make sure I'm clicking the right things. There we go.
Dude, I know.
Um, So let's see. So convergent evolution is, I think, definitely one of the more interesting features that can happen during evolution. It is when things that are not particularly closely related to each other happen to stumble on similar solutions to similar problems. And this shared feature that they have is not a result of their shared ancestry. So a classic example of this is the camera eye in vertebrates and in cephalopods. The ancestor of these two clades didn't have a camera eye. It had very, very simple eyes. And yet, these two very different groups of organisms independently have evolved very similar lens-based complex eyes. And so this is kind of a classic case of convergent evolution. These two things that are not related have a very similar feature that they have evolved over millions of years. The interesting thing about that is that if you look at the genetics underlying how the eye is developed in these taxa, it actually uses the same genes. Most of this is controlled by the PAX6 gene. There are several other genes that are being found out right now that are used similarly between these two clades. And they've both figured out ways to co-opt the same genes to come up with very similar solutions. And so this is something that's kind of happening more commonly as we learn more about molecular development, and genomics, we're learning that a lot of these classic cases of convergent evolution are better described as what's called parallel evolution. This is still an independent evolution of similar features in distantly related taxa, but they're using similar starting places. They're using the same genetic or developmental toolkits. And so it kind of gets in this weird zone of like, it's kind of a result of shared ancestry, but not quite, because they're using the same genes, the same precursors to do the same thing, but they've figured it out independently in very distantly related taxa. So I find this really fascinating, and I am very interested in learning how many of these cases of convergent evolution are really parallelism if you look deep enough, and how common is parallel evolution. This is important for a couple reasons. So my main research questions that I look at in my work, first of all, how common is parallel evolution? Like I said, that's going to be the question that we talk about most today. I haven't quite gotten to looking at the other things in my research. But other things that are important to consider here is, what kinds of taxonomic levels does parallel evolution happen on? Looking at the human and octopus example, those are, across huge, like phylum level differences between those clades. But does this happen more commonly at like the family level or the genus level or, the kingdom level? Like what levels does this tend to occur on? And the other thing that I think is most pertinent for paleontologists is how does parallel evolution affect the accuracy of our phylogenies that we're putting together based on morphology? If closely related species are using the same genetic precursors and can, in parallel, evolve very similar traits, that makes it a lot harder for us to put together accurate phylogenies. So being able to characterize how often this happens and how big of a problem it is for us is really important if we want to get a better understanding of how things are related in the fossil record. So, my kind of taxon of choice in studying this question, at least of my modern subset of taxa, is in squamates. So, squamates are lizards and snakes. They are extremely diverse and have a ton of really great examples of parallel evolution within squamates. Things that are seemingly very complex, like live birth, have evolved in parallel or at least convergently in a lot of different squamates. They also have like very similar differences in their teeth, which will be the main focus of this talk. Loss of limbs. There are all kinds of things that evolved convergently among squamates. They're kind of a hotbed for convergent evolution, honestly. And we don't know if a lot of these things are parallel evolution, because that hasn't really been studied in detail. But we know that these guys are really evolutionarily labile. And so they are a good place to kind of look if we want to study how often parallel evolution actually happens and kind of how it happens. So the feature that I am interested in looking at under this lens of parallel evolution is heterodonty. So this is a difference in tooth size and or shape across the jaw. This is very obvious in mammals and is commonly cited as a hallmark feature of mammals. So, we've got our incisors, our canines, which are a little wimpy on humans, but it's still there. Premolars and molars, very distinct regions of different sizes and shapes of teeth across the jaw. And they're all specialized for their own different functions. So, here's another fun example of a crab-eater seal with these crazy premolars and molars in the back with their little cusps on them. But so this is a very common feature seen in mammals. You don't see it as often, or at least classically, we haven't really considered it as being present as much in other non-mammalian vertebrates. So I'm going to take a little aside here to talk about how did mammals get to that point? Where did heterodonty come from in the mammal line? So we actually have a really good fossil record showing where heterodonty came from in mammals and kind of the gradual changes that happened in their jaws. So we're going to walk a little bit through synapsid evolution. This is the other main group of animals that I study. I've done a lot of research with synapsids, and they tell some really cool anatomical stories in their fossil record. So our kind of starting place in this story, if we're looking at where mammals come from, is in these early sphenacodontids. Dimetrodon is like the classic example of this. And even this early on, you can see there are subtle differences in the shape, not so much the size rather. There are subtle differences in the size of the teeth, maybe a little bit in terms of the shape with kind of the longer, thinner teeth towards the front in the canine area, and then these kind of shorter, blunter teeth towards the back. So there are some very subtle differences even in this really, really ancient animal along the mammal line. And so these guys are often described as having caniniform teeth because, the people naming them didn't want to quite give it the title of canine because it's not quite there yet, but it's a caniniform tooth in these sphenacodontids. This heterodonty that you see in sphenacodontids gets more embellished as you get into things like therapsid grade synapsids. So this is an example of a Gorgonopsian. They're known for having these really enlarged saber-like canine teeth. And so very distinct regions of teeth in those guys as well. There are several therapsid clades that have an enlarged canine like this. And so it's a lot less subtle than in the Dimetrodon, but still relatively simplistic. They're still all more or less cone-shaped teeth. So as we get into cynodonts, this is where you start getting a little bit more complexity in the type of heterodonty that you see. So here's a Thrinaxodon CT scan showing, again, that really pronounced canine tooth. It's not quite, we wouldn't just call it a caniniform at this point, but like that's a straight up canine tooth. More like kind of thin, small incisor forms at the front, and then these kind of like wider, a little bit multi-cusped you can see in there on some of these molariform teeth. So they're starting to get not just differences in size of the teeth, but differences in shape, how many cusps they have on each one. So that's starting to happen in this cynodont grade taxon. And then if we get up to mammalian forms, this is where you get just like completely heterodont dentition. You're starting to get very complicated teeth. Eventually you start getting more like tripasphenic molars showing up in the back of the tooth row. This is when like you've reached full mammalian style of heterodonty. And so this has been kind of embellished in mammals to do all kinds of different things nowadays. But we have a really great fossil record kind of showing how we got to that point over the course of hundreds of millions of years. So the interesting thing is, I kind of showed you that Dimetrodon, and you could see kind of the subtle heterodonty you see there. We do have heterodonty in non-mammalian vertebrates as well. So on the right here is an example of an agamid lizard. And you can see here, it's kind of reminiscent of some of those early therapsids where you've got like a very pronounced caniniform. Again, the people naming these things didn't want to quite call it a canine because, it's not exactly the same thing. But we've got a caniniform tooth there on the top and bottom row. We've got kind of those sharper incisiform teeth, and then we've got those very like ridge-like triangular teeth in the back. And so it very much feels similar to what mammals have and what they had kind of early in their ancestry as well. And so this kind of piqued my interest because it made me wonder if maybe this is kind of a case of one of those parallel evolution type things. Is there some kind of developmental underpinning that is causing different species to have teeth of, particular shapes and sizes in different parts of the jaw? And heterodonty is pretty common actually across different vertebrates. You see it in quite a few fish. There's been a lot of study on that recently. A lot of squamates have it to some extent with either things like multicusped teeth in the back of an anoles jaw or the really distinct fangs of snakes. In things like tegus, they have really bulbous teeth in the back because they're doing a lot of crushing of invertebrates. And even in reptiles that have more subtle differences in their tooth row, you can still see differences. So this is a Komodo dragon, for example, and you can see the teeth in the back do have a slightly different size and shape than the teeth in the very front. And so even in kind of subtle ways, there do seem like there are differences across the jaw. And it's never really been studied why that is the case in non-mammals. And so in studying why a morphology exists, I always like to go back to the classics with what is termed Silaker's triangle. This is supposed to be kind of a framework for understanding different things that can cause a morphology to look the way that it does. So in Silaker's triangle, there's of course the adaptive corner. There's, this is an adaptation for a specific thing. It is the way it is because it helped the animal do something. But then there are other factors, right, that determine what a morphology will look like. There's the phylogenetic corner, that is kind of the history of, they just happen to inherit genes that do this thing, and so it just does that thing in this modern animal. Then there's also the constructional corner. This is kind of just physics are physics as a rule. Sometimes, if an animal is too big, it can't fit like a certain size GI tract in it or skull or whatever. And so that would be another reason that you might see a certain morphology in a species. So really here what I'm interested in is kind of looking at how these different aspects influence heterodonty, like how much of it is some kind of developmental signal versus how much of it is adaptive specifically to each clade, how much of it is just because of, physical forces on the teeth that might cause them to look different. And so really my research is trying to understand how does heterodonty get influenced by these different factors? And does that tell us if it's actually parallel evolution or not? And so in trying to figure out why do reptile teeth often have this kind of like semi-heterodont dentition? Why do they follow a specific pattern? There's a couple different hypotheses that I'm kind of looking into with my research. First of all, for mammalian heterodonty, we do know why that develops the way it does. It is caused by genetics. There are certain Hox genes that kind of regionalize the jaw and cause downstream changes in gene expression. So there's different regions of gene expression that demarcate the incisors versus the canines versus the premolars and molars. And so one like obvious idea for where reptilian heterodonty might come from is since we're seeing kind of similar regions in reptiles, maybe there's some kind of developmental signal that just happens in all vertebrates there. And maybe that's what's eventually being co-opted to form different regions of the tooth row. So that's one hypothesis, is maybe there's some kind of shared developmental signaling going on here. And this would be the case in which it would be considered parallel evolution specifically. There's also other possibilities though. So one is that different teeth along the different positions on the jaw are going to be exposed to different forces. So just because of how lever mechanics work, the closer you are to this kind of fulcrum of where the jaw hinge is, the more force that tooth is going to be experiencing. And so teeth do respond to forces when they are developing. And so it's possible that maybe these differences in the size that we see along the jaw are purely a result of different forces causing the teeth to just grow differently. This would be kind of more in that constructional corner of Silaker's triangle. And then another thing to consider with reptiles is that they continuously replace their teeth. Mammals are what's called diphyodont, which is where we only get one set of replacement teeth for the most part. And that's the adult dentition that we live with. But reptiles are continually replacing their teeth. And so there's also a factor to consider there, where as the reptile is getting larger with its life, it will just make bigger teeth. And so teeth that maybe aren't getting replaced might stay smaller just because they existed when the animal was smaller. So this would kind of also go into, I would argue, that constructional corner of the triangle. So the goals with my research, really in looking at this question of heterodonty within squamates, is to explore, first of all, how common it is. Because like I said, heterodonty is usually considered to be a mammalian feature, and oh yeah, there are like some reptiles that kind of do it. But people haven't really looked at, what does it look like more subtly in the jaw in some of these reptiles? Like how common is heterodonty on different levels in squamates? And then additionally, I wanted to characterize the regions of the tooth row in squamates to try to start understanding what mechanisms might be generating those different regions. So just a second, we got to Cool, the thing is covering up my thing. So my first question to look at was just how common is heterodonty among squamates? So what I did for this little part of the project was I just looked at all the different squamate skeletal specimens that we had at the Peabody Museum at Yale, which is where I was when I was conducting this research. And I was kind of just going through qualitatively assessing the degree and the type of heterodonty. So like, is it really, really clear demarcations of tooth regions like we saw in that agama? Or is it more subtle like the Komodo dragon we saw? And so as I was doing this, I quickly realized that heterodonty is actually more the rule than it is the exception. The vast majority of the squamates that I looked at least, had at least some subtle heterodonty in the size and or shape of the jaw. The only taxa that I really looked at that didn't have any indication of heterodonty at all were a couple of geckos. Everything else had at least some kind of size difference or something along, the jaw front to back. So an example of this would be like, you can see really subtle examples, like in this Gila monster. And so this would be an example of kind of size heterodonty, where you've got really small teeth in the front there as the kind of incisiforms, if you will, and much longer teeth towards the back of the jaw. So that would be an example of just size heterodonty. But there were also examples like in this iguana where you don't really get differences in size across the jaw. Mostly it's differences in shape. So you kind of get more conical teeth in the front of the jaw where it's kind of that incisiform region. And then the molariform had this really distinct leaf shape to them with all of these little mini cusps. And so this made it very clear to me that, this is kind of something that has been overlooked for a long time and is ripe for further study. Just why so many of these squamates have these differences across their jaw. So the next step that we are currently working on is trying to look at what the actual regions are in the tooth row, because especially if we go back to like the iguana here, they kind of grade into each other, those conical teeth versus the leaf-shaped teeth. And so we wanted to try to quantify where the differences in the regions are, and a great way to do that is with geometric morphometrics. So using this, you can place landmarks on surface scans, or we basically took like the surface files from CT scans of all these different lizards, and you can put landmarks on them to try to calculate kind of the differences in the distances and get an overall estimate of the shape along the jaw. And so those quantified shape differences can then be used in what are called segmented regressions to kind of see where like inflection points in shape changes are happening along the jaw, where they're starting to follow different patterns. And so there have been people who have done stuff like this to a kind of limited extent. So this is an example of some data from a study on, I believe this was in crocodilian teeth. So you can get kind of like an idea of, as you go from like the first pre-molariform to like the back like molariforms, like what are kind of the differences along a principal component axis? What are the kind of shape differences that are being summarized, and how do those change over the jaw? And so we kind of wanted to do something similar with this, but use 3D landmarks because it would give us a little bit more data to work with. Sometimes, if a tooth is kind of like bulbous, you're going to get that shape on all sides versus if it's kind of more of like a ridge, that would also be different. So we wanted to really capture the 3D aspect of the variation that we were seeing. So we applied this to 3D landmarks, CT scanned individuals of a couple different species. We've now gotten more species than apparently the last time that I updated this slide, but we haven't quite gotten results yet for those guys. And so we put landmarks kind of at like the base, in different parts and on the apex, and also used some sliding semi-landmarks along the edges of the tooth and the base of the tooth, just to kind of capture overall, like, a tooth is a fairly simple shape, but we wanted to kind of capture different types of curvature and different widths and stuff like that, and rounding. So here, I'll show you kind of the results that we have for the two species that I've really gotten good data on so far. Um, so this is that agama lizard that I showed earlier. This one has just by looking at it, you can see it's very clear heterodonty. As I said, it's very reminiscent of a kind of mammalian pattern. So we were very interested in kind of looking quantitatively to see, kind of use this as a test. Like, this has very obvious heterodonty. If our method works at uh, identifying heterodont dentitions, it should be able to pick up on this. So we placed our landmarks and did our analysis, and we found that it kind of gives us three regions. So it shows us that there's a distinct region in the front of the teeth. You can see in the graphs here, this is kind of showing the different principal components as you go along the position of the tooth along the jaw. So somewhere around kind of tooth position 3 or 4, there's kind of a change in how the principal components are related to each other. So you can see it kind of picks up on like this incisiform, like cuneiform region in the front. There's maybe like a premolar slash molar thing going on. So kind of three distinctions back there. So at first we were like, oh, that's really interesting. Like it kind of looks like, premolars versus molars. But one thing that was important to note is that these teeth kind of sequentially get bigger as you go towards the back of the jaw. You can kind of visually see that as well. And so one thing that's important to note there is agamid lizards are interesting in that they don't replace the teeth in the back of the jaw. They replace the ones in the front, but the ones in the back actually stay there and they are fused to the jaw throughout their life. And so this is kind of showing another way. This is like with little box plots, just kind of showing where each tooth is on the PC axes. And so you can see again, the three teeth in the front are very, very distinct. And then there's kind of like ebbing and flowing, very subtle differences towards the back of the jaw. But if you look at a small, juvenile agama, they have really small teeth because those are the teeth, they kind of, they're the teeth that were around when they were juveniles. They were smaller animals. And so as the animal grows, they add more teeth to the back of the jaw. And so this could easily just be a case of, that premolar slash molar transition could be some kind of a growth spurt where maybe the animal was pretty small at first. And then as it is physically growing more, the teeth are getting bigger towards the back of the jaw. So not some kind of developmental modularity thing like you'd see in mammals, really more of a constructional thing of this is just a result of the animal getting bigger over its life. So that seems like, it doesn't totally rule out a developmental cause for it, but it does seem like, it's important to take tooth replacement patterns into consideration as you're looking at these scans. So the next animal that we wanted to look at was kind of a more subtle example of heterodonty. This is in Varanus monitor lizards. And you can see it just from looking at the scan. They kind of have these longer, thinner, recurved teeth in the front, but then they get more bulbous teeth towards the back. And so it's more subtle heterodonty. It also seems to change with ontogeny. Those bigger teeth tend to come in when the animal is an adult. But it does continually replace its teeth, so it's not going to be just a consequence of the animals getting bigger over time. All of these teeth are getting replaced consistently over the life of the animal. So we wanted to compare our 3D results with some 2D results that were already done on this animal. And we wanted to see, again, kind of testing our methods to make sure that we're actually able to capture subtle heterodonty that we can see with our human eyes, but being able to quantify it with the computer program. So we placed our landmarks, and this time we saw kind of a change in, what was it, like tooth position 11, kind of towards the back of the jaw. And this makes sense because that is kind of where the teeth tend to get more bulbous towards the back. And so it seems like our model did a good job of recognizing where those delineations were in even a more subtle example of heterodonty. So this is what that box plot looks like. You can see this one is really, really subtle, but it does start to have kind of some different variations in the back of the jaw, especially in that first principal component. And so this one is great because that this one is not a result of tooth replacement because their teeth are being continually replaced over time. There's no reason to think the teeth at the front of the jaw would need to be any smaller than the ones at the back of the jaw. So it kind of rules out that aspect of that kind of constructional corner, and then begs the question of, is there some kind of developmental mechanism, some kind of signal that is different between the front and back of the jaw that tells the teeth to develop differently there? Or is it maybe a result of like forces, stuff like that? It's kind of a big up in the air question right now, but definitely something to look into more. So the conclusions that we have from this study so far are a little bit limited. But first of all, we were able to conclude that our 3D geometric morphometric approach is pretty good at identifying even subtle regionality in squamates. And so that gives us a good jumping off point for future studies looking at other reptiles that are even more subtle. Like, how subtle can we get before it doesn't really pick up on things anymore? And so that's something we're working on right now, is trying to get more landmarks placed and more taxa studied. The other thing is that heterodonty does seem to be the norm rather than the exception in squamates. Sometimes it's just based on size, sometimes it's just based on shape. Every once in a while you get really clear size and shape differences. But it does seem like squamates in general have a lot of heterodonty that kind of hasn't been appreciated in the past and is definitely ripe for studying what mechanisms are there. And then it seems like the heterodonty is caused by kind of a variety of factors. We don't know necessarily yet if there is kind of this underlying developmental factor that's uniting all of them, but we do know that it's really important to keep in mind the replacement pattern that you see in the teeth and kind of those more constructional things based on the animal's size. Yeah, so agama seems to be driven by a combination of developmental factors and a lack of tooth replacement. It is important to note that like even within one of the teeth in the front of the jaw in agama, the ones that do get replaced, there is still that big caniniform tooth. And so that does still kind of say, that's not a thing that's just from tooth replacement and jaw growth. That might be some kind of developmental signal that's specific to that caniniform tooth. So kind of a combination of factors in that taxon. And then in our monitor lizard, it seems to be probably mostly driven by developmental factors, or it could be forces in response to feeding, which would have to be kind of ruled out also. So the future things that we are trying to do in my lab right now, one is, like I said, trying to analyze more squamate taxa to try to test the sensitivity of our methods. How subtle can you get before we aren't able to get a signal anymore? And then what can we do with these quantifications once we get them? We also are interested in trying to investigate possible developmental mechanisms. I've done a little bit of embryo work to try to look at these things, but it's kind of slow going because you got to wait for the lizards to lay their eggs. So we'll see if we ever get that program off the ground. We're also hoping to eventually apply this method to heterodonty in synapsids, because like I said, we have that great transition from mildly heterodont things like Dimetrodon to a fully heterodont mammalia form. It would be really cool to kind of quantify this over the course of synapsid evolution and see where were the real big changes that happened. Another thing that we're working on that I didn't really put on this slide is looking at the replacement pattern in these different lizards that we're looking at, and trying to understand really how might that be affecting the differences we see in the tooth shape along the jaw. So with that, I'd like to thank my funding agencies and my coworkers, and I am happy to take any questions.
Thank you, Savannah. This was an excellent talk and very exciting material. I would like to remind our audience that if you would like to ask a question to Savannah, now is the time. You can drop your question in the chat box or send it to Nina, or you can just raise your hand and we'll let you unmute and you can ask a question. And the audience is also sharing their excitement of the talk through Zoom reactions. So Gil's Kani has asked a question, and I'm posting it in the chat. In therapsids, are the caniform tooth the first tooth on the maxillary? if I'm not mistaken, in modern animals, the definition of a canine is more related to its position on the upper jaw than to its shape. And then there is a related question where he's asking, so squamate heterodonty on the upper jaw is not linked to the separation between premaxillary and maxillary.
Yeah, so that's a good question. So yeah, in mammals, it's defined as it's always kind of where the maxilla and premaxilla meet. In synapsids before mammals, it does seem to be generally in that area. It's not always exactly at that spot, but it does seem to be associated with that region at least, which does imply that there might have been some kind of developmental difference that kind of happened at that part. In reptiles, it seems kind of like the therapsid version, where it's more or less in that position of being where the maxilla and premaxilla meet, but it's not exact, which is another reason why I suspect at least there might be something developmental going on there. Especially since, the Hox genes that all of this relies on are pretty conserved in the facial region. And so I wouldn't be surprised if it's kind of, the premaxilla and maxilla ending where they do and the teeth being regionalized where they are is probably all relying on some kind of earlier signal. We just haven't really confirmed that because we've only ever looked at this in mammals. And, our two main developmental study organisms are mice, which are mammals and also have a weird diastema, and chickens, which don't have teeth. So we really don't understand that well in terms of the comparative aspect of this developmentally.
Thank you. Um, we have an anonymous question, which is asking, you mentioned that some geckos don't show any heterodonty. So, do you think that has some functional reason, for example, diet or just evolutionary coincidence?
Yeah, I haven't really given a lot of thought to that. It is interesting. Sorry, the sun trying to figure out how to not be burning. It is interesting that like this is kind of the rule is that there are some differences, but there are some taxa where it is pretty much the exact same across the tooth row. And especially since, like I said, like the back of the jaw should be exposed to different forces than the front of the jaw. And that's just a physics thing. That's not going to depend on the taxon. That's just it's closer to the hinge. It will have higher force. That is how levers work. So it is interesting that some geckos don't really have any differences there. And I haven't really thought of a good reason why that might be. If there's like a reason they would need that, because I mean, if you think about it, most of them are eating bugs and stuff like that. I don't know how important it would be to have specialized teeth in the front versus the back for something like that, or like, it seems like the specializations would be more useful than having them like pretty much identical along the tooth row. So I don't know if there would be some kind of selective pressure there, or if they're maybe they just have some kind of developmental difference. What it kind of reminds me of is cetaceans, so toothed whales, dolphins. They also have like pretty much perfectly homodont teeth front to back. Like there's not even really size differences there. And so there is some change that's happened developmentally that's caused that in them. Maybe it's possible that that's happened to geckos as a result of some other thing. We just don't really know enough about their tooth development to know what that would be.
Mm-hmm. That's fair. Um, we have another question, which is asking, what do the growth stages for teeth look like? Do they all start out from the same blueprint, then develop into different shapes as they grow, or is there a difference from the start?
Yeah, that's a good question. I mean, it's interesting because they're being replaced over time, and so we don't really know. I'd have to kind of look at like one tooth position and kind of see how it changes as like an animal gets bigger, which would be really interesting to look at. Um, it does seem like at least in the varanids, because like I was saying, they kind of have like the adults will have more bulbous teeth in the back, but the babies will have kind of more pointy teeth in the back. And so that is a result of like, the first teeth that develop there when they're replacing them when they're little, they're kind of pointy. But then as they get older, they start making more and more bulbous teeth when they replace them, which is really interesting. And like I said, there's a lot of different factors for why that could be. But it does seem like as they replace them over time, sometimes they do change.
Yeah, thank you for that answer. We have another question anonymously. Have you tried to quantitatively compare the degree of heterodonty across the different life stages of squamates?
Yeah, we haven't really done that yet. I think that would be really cool, especially, I mean, I guess to some extent that's what the regressions we do are accomplishing because I try to like have different sizes of animals in that data set. And so it's kind of looking at like, how do the teeth grow within each of these positions? And so it's kind of subtly getting at that, but it's not telling us a whole lot of really detailed information. It would be cool to kind of look and more like qualitatively describe how the teeth are different across the jaw in like larger and smaller animals. But that's not really something that we've done yet.
Mm-hmm. Yeah, yeah. I had a related question to that. Like, you know, it seems like we know more about teeth development in mammals than in reptiles. So, like, how are you kind of preventing, this bias of understanding teeth development in reptiles? Uh, because, like, we are mostly being informed from what we know about mammals. And, like, yeah, how does that, show up in your analysis or, like, yeah.
Yeah, I think that's something I've definitely had to try to intentionally think about because, like I said, when I started this project and had this question at first, it was very much me being like, oh, that looks like mammal. Let's see why it looks like mammal. And maybe it has the same developmental pattern, like trying to apply these rules that we see in mammalian heterodonty to a reptile version. And it's been really important for me to step back and go, okay, their teeth are very different from ours. Like they're replacing them constantly. They're adding them onto the back of the jaw as they get older. There are just kind of different rules involved in making a reptile tooth. And so I've definitely kind of had to step back and think about those more and really thinking about what other factors need to be considered here. What are the other things that could be causing this to be similar between them?
Yeah, and hence maybe the projects for like looking at the developmental stages of a lizard. Like that would be really cool if you managed to do that. Thank you for answering that question. And we have another question, and this may be our last, unless someone else from the audience has more questions. Um, would preservation be an issue if you want to apply this method to fossil squamates?
Yeah, I haven't thought too much about working on fossil squamates. It's like the taxa that I've worked on so far in my career are uh, extinct synapsids and modern squamates. And I haven't really done like modern synapsids or extinct squamates, so I haven't really crossed those lines yet. Um, but in terms of fossil squamates, it could be difficult because a lot of the teeth tend to be missing. One thing that's nice about the analysis we do, because even in modern squamates, a lot of times the teeth are missing. They're constantly replacing them, which means teeth are constantly falling out. And sometimes you have like a partially developed replacement tooth. So the analysis we do takes into account that there can be missing data. So you can tell the program like, hey, this tooth is missing. You can kind of, um, estimate this so that you can finish the analysis, but then keep in mind that tooth isn't real. So we'd still be able to probably apply it to fossils. It might be a little more difficult if half the teeth are missing, then that might not be super usable. But if it seems like a relatively complete jaw, it shouldn't be too bad. The only issue there would be like, if a whole segment of the tooth row in the back is missing, or for a lot of these taxa where you only have one specimen of a species because it's some tiny little lizard that is kind of understudied, those wouldn't really be useful because, like I said, this was done with like at least 20 individuals of each species. There aren't a ton of fossil lizards that you could do that with, especially because their teeth or their jaws fall apart so easily.
Yeah. Um, yeah, I was wondering what, species or like animal would be suitable for doing this kind of parallel evolution study. Like, do you have any newer species in mind or like, group of um, animals you want to try this on?
In terms of the development.
Yeah, like in terms of checking parallel evolution.
Yeah, I one thing that we tried doing when I was at Yale was looking at bearded dragons because one, they're really easy to find because you can get them at any pet store. Um, and they're pretty easy to care for. And they usually do a pretty good job making eggs. I didn't have I was only there for like 2 years, and so getting like the breeding colony set up and then trying to troubleshoot how to keep the eggs from like spoiling before we found them or getting stepped on because they bury them and then sit on them, which is like these guys have like no survival skills. They're like purely pets at that point. But that would be a great animal to study at some point if I can get them to lay eggs properly. Maybe I just need to make friends with somebody who has a working bearded dragon colony. If anybody knows someone, I know there are people who do some developmental studies with them, but I kind of don't want to because it's honestly just a pain trying to work with live animals. I miss the fossils. They're dead. I don't have to feed them. Um, yeah, so I tried working on this with bearded dragons. There are several geckos that would be good to kind of use as sort of a control since, like I said, they don't really have a lot of heterodonty in their dentitions. So that would be cool to look at. Um, and then there's other like pet shop style lizards, like anoles have like some level of heterodonty, tegus do,
Hmm.
chameleons. The main issue is like the husbandry with those animals and getting them to actually lay the eggs.
Ah, well. Um, with that, I would like to thank you again, Savannah, for an excellent talk, and to our audience for being here. And we will be having a seminar next Tuesday. If you want to join us, please do. By a seminar by Leticia Resande from University of Santa Maria, Brazil, and she'll be talking to us about Triassic Archosaur paleobiology. And yes, next up we have tea time, which is an informal session, relaxed setting to talk to our speaker. And before that, we'll take a quick 2 minute break. Um, and you can stretch, roam around, get your paleo pets, and we'll be back soon. Bye.